This lecture is part of a series of lectures for Rad229, MRI Signals and Sequences, offered in the Department of Radiology at Stanford University. The 15th lecture, Magnetization Preparation Part 2, focuses on diffusion and is broken down into 3 parts. Lecture-15C covers the advanced diffusion imaging methods. The learning objectives for this lecture include being able to understand the importance of the diffusion encoding gradient direction, to appreciate that the diffusion tensor has an eigensystem that relates to the underlying microstructure, to explain the effect of bulk motion on the diffusion signal, to describe how gradient moment nulling can be combined with the diffusion encoding to limit the impact of bulk motion. You'll recall from the previous lecture a diagram that looked just like this, that highlighted that during the spin echo encoding sequence, we can add 2 diffusion encoding gradients, and we can repeat 2 experiments. One experiment without the diffusion encoding gradients, which allows us to obtain the B0 image, the non-diffusion weighted image, and a second experiment with the diffusion encoding gradients turned on with a B value, say, of 1,000 seconds per millimeter squared, and that allows us to obtain a diffusion weighted image. Mathematically, we can use these 2 images together to calculate the underlying diffusion coefficient and produce an apparent diffusion coefficient map. Now, one thing we can also control in this experiment is the diffusion encoding gradient direction. So we also saw in the previous lecture that we could change the magnitude of the diffusion encoding pair to be any linear combination across the slice select, phase encode, and frequency encoding directions. So what's shown here on the right-hand side is a set of directions, 6, maybe 8 different directions that are playing out in sequence, and we can encode diffusion along any of these individual directions. And this effectively gives us a projection of the underlying diffusion process along the particular gradient direction. And if you look carefully at the images here, you'll notice 2 things. One is a feature we talked about in the artifacts lecture, where we can see subtle distortions of the images between the different encoding gradient directions that arise as a consequence of eddy currents. The other thing that you'll also notice, however, is that the underlying image contrast itself is changing. And this is a really fascinating observation. It means that the direction of the diffusion encoding gradient, the only thing that's changing during these sequence of images, changes the underlying diffusion image contrast. And that's because we're sensitizing ourselves or sensitizing the experiment to a projection of the diffusion tensor within each individual pixel. And because there's significant microstructural organization in the brain that varies over space, this gives rise to significant variations in image contrast as well. So we can expand on the diffusion encoding signal expression that we had previously to include both the diffusion weighted image, the non-diffusion weighted image, and this time we also include a constant B value, so we take this as a constant, but we specifically denote the gradient direction. Now, because D is a 3x3 tensor, we need to multiply in 2 different ways the diffusion tensor onto the gradient vector direction and then dot it again with the gradient vector direction so that we have the right projection of the diffusion tensor in the end. But ultimately, we can do a series of experiments where we acquire several different gradient encoding directions sufficient to actually allow us to estimate the underlying diffusion tensor. So here again, we see the expression for the simple diffusion weighted imaging experiment. And here we compare the diffusion tensor imaging experiment. Now, it's not yet obvious that you could use this encoding information to extract the diffusion tensor, but we'll describe some of the details moving forward. So again, Sj would represent the image signal intensity with each of the diffusion weightings, and it has a specific image pixel signal intensity that depends on the fundamental diffusion tensor itself, as well as the gradient directions of the diffusion encoding gradients. We measure S0 as before in the absence of the diffusion encoding gradients. We have a fixed B value for this experiment, so that the only thing we're changing is the gradient directions. And the gradient directions would be just unit vectors pointing any number of different directions. Now, it turns out there's some pretty sophisticated and judicious ways to choose the set of directions that might be good for your experiment, but that's not a topic of this particular lecture. And finally, if we've done all of those experiments, we've done J experiments collecting SJ images as well as the S0 image, mathematically we can invert this expression and actually estimate what we call the diffusion tensor. Now the diffusion tensor tells us about the underlying diffusion process and the magnitude of diffusion along, say, the primary eigenvector, which is the principal direction of diffusion, as well as two other eigenvectors, the minimum eigenvector and the intermediate eigenvector. And each of these eigenvectors has associated with it an underlying diffusion coefficient as well, and taken together they collectively describe the diffusive process, the anisotropic diffusive process within each individual pixel. And this is really kind of a remarkable thing, meaning that we can estimate things about the microstructural compartment within a pixel. This isn't a measurement that's made between pixels, but yet within a single pixel, or all pixels within an image. So the diffusion tensor itself can be represented by a set of eigenvectors which tell us something about the orientation, that is the orientation of the microstructure within that pixel. And it can also, the diffusion tensor also conveys some eigenvalues. And those eigenvalues can be taken individually to tell us about the maximum diffusion along, say, a particular direction, the primary eigenvector direction. That would be the direction within the microstructural element that has the fastest rate of diffusion. But we can also calculate things that depend specifically on the eigenvalues themselves. We call these invariants, which are functions of eigenvalues, and they're fundamentally independent of the orientation. And this produces new and interesting forms of image contrast that aren't available through other means. Just an array, a range of examples is shown across the bottom here that show great microstructural detail extracted from the diffusion tensor imaging data. So here's one poignant example. This is a T2 weighted image where you can see there's obviously a large mass on the right-hand side here. It's difficult to know sort of where's the CSF and where's the underlying brain structure. And of course, this mass in this case represents a glioblastoma. And it may be interesting surgically to understand where the white matter fiber tracts lie within tumors like this during resection surgery, for example. And so with diffusion tensor imaging, you can actually highlight quite significantly regions of structural integrity, these here being the white matter fiber tracts within the brain, and perhaps an important structure here running right through the middle of this tumor. So this could be really useful for planning surgeries to resect this tumor. In this particular map here, a few things are happening. One is we're highlighting areas of high anisotropy, that is areas that have one direction of dominant diffusion, as opposed to areas like CSF that have no dominant direction of diffusion. They just, water molecules diffuse equally in all directions, so they appear dark. And then the direction itself tells you about the dominant direction of diffusion. And so here we can see that within this structure here, this is a highly anisotropic structure because it's bright, and it's oriented generally along the y direction. That's the up-down direction in this figure. Whereas other structures in the brain are left-right oriented or even in and out of the plane in their orientation. Now the applications for diffusion tensor imaging are quite broad. I'm personally interested in the heart. This is a very challenging organ in which to do diffusion tensor imaging. The origins of this work actually start with trying to do diffusion imaging in ex vivo hearts because fundamentally there's a lot of bulk motion within the heart, and that bulk motion, if not accounted for very carefully, can completely corrupt the underlying diffusion measurements. So why might we want to look at diffusion in the heart? Well, similar to the brain, there's a great deal of structure and structural organization that the function of the heart depends upon. So we're interested in developing diffusion methods that allow us to map out microstructural organization in the heart. But to do so, you have to overcome several challenges, the obvious one being the significant bulk motion of the heart. So why is it that motion corrupts the diffusion-weighted imaging signal? Well, here we have a simulation of flowing spins and a conventional diffusion gradient encoding experiment. So this is the simple bipolar experiment where you have a large positive gradient followed by a negative gradient. And if we back up and play the movie again, you can see that as the spins are both flowing and there's underlying diffusion, that the signal will drop substantially and it never will recover. That by the time you play the rephasing gradient, the combination of diffusion and flow leads to substantial phase dispersion across all of the spins, and their coherent sum is basically 0 at this point. So there's no good way to recover signal from a diffusion-weighted experiment in a spin system that's flowing. So what's the alternative? Well, it turns out you can actually motion compensate your diffusion-weighted imaging gradients. This is similar to motion compensating gradients that we discussed in the motion compensation and motion encoding lecture. So here we can change the gradient waveforms that we're using. These gradient waveforms will in fact have, say, first and second moment nulling, but they'll still impart diffusion sensitivity. And as a consequence of this complex motion coupled with diffusion, you'll go through a period of dephasing the signal, but at the very end, you're still able to rephase the signal. And this is really rather remarkable when you think about it, right? Let's step ahead here again. So watch how the phase is being printed on the spin system. Almost, it's very patterned, but it has so much intravoxel phase that its coherent sum will be very, very low. And yet at the very end, this last part of the gradient is able to rephase the gross majority of that signal and still give rise to a signal that would then be diffusion sensitive. And so we'll discuss the mathematics of this a little bit further. Here's just another example of demonstrating sort of the advantages of adding motion compensation to the diffusion-weighted experiment. So the conventional diffusion-weighted imaging experiment, just the monopolar spin echo experiment, two gradients, one on either side of the 180, leads to substantial signal losses. And so you can see as this person's moving their head during the diffusion encoding experiment, there's entire imaging volumes that are completely lost. Whereas on the right-hand side here, we've added motion compensation, that is moment nulling, to a diffusion encoding experiment. And even though this patient or subject is moving their head substantially, there's quite a bit of signal present in most of the imaging slices. So while this doesn't solve the particular problem for a substantial brain motion or head motion like this, it certainly improves the situation. Okay, so let's look at the diffusion-weighted spin echo EPI sequence again. We've got two diffusion encoding gradients on either side of the 180, followed by an EPI readout train. This is a very popular sequence. It's widely available. It's very fast because it's a single-shot spin echo EPI sequence, but it's also very bulk motion sensitive. And in fact, there's a little bit of wasted time before the 180 here because of the timing required to reach the middle of K-space and the middle of the EPI readout, plus this diffusion encoding gradient. The timing calculations are such that there's some time here that's available, so to speak, or wasted. And in fact, this sequence has relatively moderate signal to noise and moderate spatial resolution. So the objective of what I'll describe now is to develop bulk motion insensitive diffusion-weighted imaging sequences to minimize the echo time as much as possible and to work towards having shorter scan times with acceptable signal to noise and perhaps higher in-plane resolution. And of course, a key ingredient for this will be convex optimized diffusion encoding, and it helps if you have high performance gradient systems. So this slide we saw earlier in the gradient waveform design lectures, and we described time optimal gradient design as a convex optimization problem for which efficient solution methods exist. So we can start with the timing for a conventional monopolar diffusion encoding sequence for which we know there's some wasted time. We can take into consideration gradient hardware, perhaps physiologic constraints, although we're not taking that into account here. And we can importantly take into account pulse sequence constraints like moments, the first moment or the second moment, and the p-value so that we can maintain diffusion sensitivity. And by way of optimization, we can design new sequences that have the properties that we want. In this case, this is a diffusion-weighted imaging sequence with a very short TE. So this is the process that we described previously in the gradient optimization waveform lecture. I won't spend too much time on this, except for to remind you that some of the constraints can include the gradient moments, and that matters to us quite a bit for this experiment because we want to minimize, say, this first moment and the second moment, the velocity and acceleration contributions, while being subject to particular hardware constraints and other pulse sequence constraints. And in this case, we're showing how we want to maximize this beta function which is in fact related to the B value. So without stepping through the details, I'll just remind you that we covered this kind of material in the gradient waveform optimization lecture. So let's remember again, or discuss again, how gradients encode diffusion. So the MRI signal phase accumulates for several reasons when you have diffusion gradients active. So the phase itself is the integral of the gradient waveform, and we saw previously that we could take a Taylor series expansion to include initial positions, velocities, and accelerations. But there may be additional gradient waveform contributions that come from other aspects of the diffusion encoding gradient waveform. And so the idea is to define a set of experiments that will allow us to account for, say, the velocity encoding, or in fact the acceleration term that's not shown here, and null those terms while preserving the diffusion sensitizing components of the gradient waveform. And in fact, this can be done. It seems like these things could almost be at odds with one another, but in fact it's not terribly difficult. So we can design, for example, gradient waveforms for which the M0 is 0. We could also design gradient waveforms for which the M1 is 0. And while I'm not showing it here, you can imagine easily doing this for gradient waveforms to have a minimum second moment as well. So let's look at an example of another example of why motion compensation is so important. So here we have a coronal image through the heart, the lungs on either side here, and below both of those is the liver. And so we don't generally think of the liver as moving very much, but during the course of a breath-held scan, in this case, the heart itself imparts a significant amount of motion to the liver. And so if you want to do liver diffusion-weighted imaging, you actually have to be aware of the fact that the heart itself will impart a significant amount of motion onto the liver, and think about ways to compensate for that. So there are a couple ways to do this. This is what we call a bipolar diffusion encoding sequence. And so you can see on either side of the 180 we have a motion compensated gradient waveform followed by another motion compensated gradient waveform. So this gives you M1 compensation. It's still fast in terms of imaging because it's single shot EPI, but this preparation time gets longer, of course, because of the added gradient lobes, and so you have a longer diffusion prep. In the case of trying to minimize cardiac motion, you could also think about adding ECG gating, but that's sort of an extra step, especially when it comes to what otherwise would be just a liver exam. So the solution that we can show that arises from the convex optimized M1 nulled sequence is that we can achieve M1 compensation as above, but we can do this with a shorter diffusion preparation interval. And this ends up being, say, fast or very fast relative to the alternative approach. In particular, when you look at the echo times, you could have significant echo time savings, which in the case of the liver that has a intermediate or even short T2, T2 star, that can be a substantial signal savings. And in fact, when you add things like motion compensation, it becomes less necessary to consider things like ECG gating. So let's look here at a comparison. This is actually a comparison so that the importance of bulk motion compensation is more clear. Here we have the monopolar sequence with a B value of 500, that's typical for liver, and the code M1 sequence, so motion compensated. And we can see that the echo times between these two sequences are similar. The TE for the code sequence is a little bit longer, but it's also motion compensated, whereas this monopolar sequence is not motion compensated. So here we can look at the apparent diffusion coefficient maps for an axial slice through the liver, and you'll notice that in this region over here, this is what lies relatively close to the liver. You have elevated apparent diffusion coefficients. That's because there's a lot of bulk motion coming from the heart pushing onto the liver. So you get an artifactually high ADC values in this region, whereas the ADC map generally flattens out across the liver when you add motion compensation. And we can look at this more quantitatively, and we can see that when we measure the diffusion coefficient in several regions across 10 individuals, that with the motion compensation technique, those measurements are more uniform across the liver regions, whereas with the non-motion compensated technique, the values themselves vary significantly more, and in fact are quite high in regions that are close to or adjacent to the heart. and there's also typically more variance across the subjects as well, because it's a less robust method for measuring diffusion in a moving target. So overall, that code approach adds motion compensation with a minimum TE penalty, or a small TE penalty, compared to the monopolar approach. So here's another possibility. You could actually add motion compensation to include first and second moment nulling. The gradient waveform itself gets a little bit more complicated. So this is a code spin echo EPI sequence with M1, M2 compensation. It does have a short diffusion prep compared to the alternatives, which are multipolar gradient waveforms. And so relative to those other possibilities, it's a relatively fast sequence. And it gives you more flexibility in your cardiac cycle timing. So now we're thinking about, if we add M1 and M2 compensation, maybe we can actually make some measurements in the heart itself. This is just a comparison of the echo times for a range of B values and EPI readout durations, just showing that with the motion compensated code sequence, we have generally shorter TEs compared to the conventional approach for doing motion compensation. So overall, the TE savings is on the order of somewhere between, say, 20 and maybe even 40 milliseconds. So there's an advantage in that the sequence is a faster way of achieving both M1 and M2 motion compensation. So here we can make a comparison again between the code sequence, which has the M1 and M2 motion compensation, to conventional monopolar sequence. Now, the conventional monopolar sequence is not motion compensated, and so when the diffusion and encoding gradients are turned on, there's essentially an absence of the heart because of the underlying bulk motion. The non-diffusion weighted image still looks pretty good, but when you go to calculate an apparent diffusion coefficient, you get very, very high ADCs because of this unrealistic signal attenuation in the diffusion weighted images. Whereas when you add that M1, M2 moment compensation, you can actually recover images of the heart quite nicely, and you still have a good non-diffusion weighted image, and you have reasonable diffusion weighted images as well. And now you can actually calculate an apparent diffusion coefficient in a moving target organ because of this gradient waveform design approach. So we can look at the numbers here again, and you can see that this code M1, M2 approach compared to the monopolar approach, it gives you a lower ADC, which is what we expect when we add motion compensation. We're getting something realistic rather than values that are faster than, say, free water. And you can see the variance also drops a lot. So the intersubject variance is quite a bit lower. And in fact, that's well shown here by looking at the ADC standard deviation. So you have higher and an even wider range of standard deviations compared to the code sequence itself. And then the code sequence, if you look at the B0 images, it has a slightly longer TE, and so you do give up a little bit of signal to noise. So it's not completely for free, if you will. So generally, this approach is a nice motion compensated approach that's generally SNR efficient and certainly more robust than the monopolar approach. So that's just a few ideas about ways in which you can advance your understanding of diffusion weighted imaging to both tensors and motion compensated tensor experiments. Diffusion, as you'll recall from the earliest lecture, can be simulated from a random walk, and you can combine that with a block simulation to better understand the signal response in different organs, for example. The diffusion MRI in general is sensitive to intravoxel microstructure and microstructural remodeling. And it's a really interesting technique that way, in that you really get at the details of what's happening within individual pixels. These advanced gradient waveform design methods can help minimize errors due to bulk motion, for example, but we've also talked about it previously in the context of minimizing things like eddy currents or PNS, and you could, in fact, rely on those ideas and concepts as well to further improve certain aspects of diffusion imaging as well. So thank you for joining me for Lecture 15 and for this series of diffusion encoding lectures. Thanks.